Hello everyone, welcome back. Today I'll be covering articular cartilage, a part 2 video. Started off with an introduction, nutrition by double diffusion of synovial fluid. Adult articular cartilage composition include chondrocytes, extracellular matrix, ECM, water, collagen, aggrecan, proteoglycan. Now let's get started. Introduction. Articular cartilage, also known as hyaline cartilage, is a unique and durable connective tissue that plays a critical role in the physiological mobility by providing nearly frictionless articulation for mechanical load transmission between joints. Owing to the scarcity and poor proliferative activity of the adult chondrocytes, a lack of vascularization and innervation, a slow matrix turnover, and a low supply of progenitor cells, the articular cartilage has a very low intrinsic self-regeneration capacity after injury. For nutrition, there is no blood vessels, no lymphatic channels, or no neural elements that enter or pass through adult articular cartilage. The chondrocytes are separated from the blood vessels of the underlying bone by a zone of dense calcification and the mature cortex of the underlying subchondral bony end plate. The major source of nutrients for the tissue is synovial fluid. Synovial fluid is a lubricant of intra-articular surface and a source of nutrients for hyaline cartilage by diffusion. This picture illustrates the articular cartilage zone. As revision previously in the first video, the superficial, middle and deep zone with the calcified cartilage. Synovial protein concentration averages about 42% of the concentration in serum. Among these proteins, albumin constitutes the single largest protein fraction, about 55 to 86% of total synovial protein, representing a major contribution to the role of colloid osmotic pressure and other physiological function. Since the synovial fluid is in itself an ultrafiltrate of plasma, whereby certain large proteins are excluded, it is apparent that chondrocytes receive their nutrition through a double diffusion system. Nutrients must first diffuse across the synovial barrier into the synovial fluid and then across the matrix of articular cartilage to reach the cells. Since it's aneural, nerve impulse cannot pass through. Any information may be because of pressure sensitive by the cells. Alteration of matrix may be caused by loading, unloading, bear load. These activities can be received by cartilage as a message to alter their sympathetic activity. This picture illustrates the articular cartilage. The dynamic mechanical stimuli occurs. There will be alteration of matrix cells by movement, whether during loading or unloading. This movement or diffusion of synovial fluid is illustrated. Adult articular cartilage composition. Normal articular cartilage consists of an extensive hydrated extracellular matrix (ECM) that is synthesized and maintained by a sparse population of specialized cells, the chondrocytes. The surface layer of articular cartilage is in direct contact with articular synovial fluid and is not covered by a perichondrium. Although chondrocytes are the primary cellular component of articular cartilage, the extracellular matrix produced by the chondrocytes imparts many of the unique functions and properties of cartilage. This figure shows the articular cartilage and the extracellular matrix molecular biomarker from the normal to osteoarthritis, and then the difference with the ECM homeostasis maintaining, then we decide the treatment. Chondrocytes, about 5% of the wet weight of the articular cartilage. Chondroblasts, which are derived from mesenchymal cells, differentiate during skeletal morphogenesis, become trapped in lacunae, and develop into chondrocytes. Chondrocytes contain the organelles such as the endoplasmic reticulum and Golgi apparatus necessary for matrix synthesis. Chondrocytes are important in the control of matrix turnover through production of collagen, proteoglycans, enzyme for cartilage metabolism. Chondrocytes surround themselves with extracellular matrix and, unlike osteocytes, do not form cell-to-cell contact. Despite their lack of direct cell-to-cell contacts, chondrocytes are still able to perform the balance between the matrix synthesis and breakdown that facilitates normal cartilage hemostasis. Chondrocyte metabolism is influenced by multiple factors, including composition of the surrounding matrix, mechanical loads, hormones, local growth factors, cytokines, aging, and injury. Chondrocytes of articular cartilage are entrapped in a low nutrient and low oxygen environment. Thus, articular cartilage has a very low intrinsic of self-regeneration after injury. Regarding extracellular matrix ECM, the ECM is a hyperhydrated tissue with values for water ranging from 60% to almost 80% of the total wet weight. The remaining 20% to 30% of the wet weight of the tissue is principally accounted for by two macromolecular proteins, type 2 collagen, which compose up to 60% of the dry weight, and the large highly negatively charged proteoglycan, Aggrecan, which accounts for a large part of the remainder. This picture illustrates the cartilage extracellular matrix. Water, around 75% weight of articular cartilage. The water content is lower in the superficial layer, approximately 65% found in the deeper layer. In maintaining the resiliency of the tissue, and it also makes an important contribution to boundary lubrication. The water content remains high throughout the life, with only a modest decrease noted with advanced age. The content of water increases with immobilization, denervation, and osteoarthritis. Since most of the extracellular matrix consists of almost equal parts of collagen and proteoglycan, it would seem appropriate that these macromolecules are somehow involved in holding the water. Collagen. 20% wet weight, more than 50% of dry weight, mainly type 2 collagen. Other types involved are type 5, type 6, type 9, and type 11. Type 9 is a short fibrillar collagen that contains a proteoglycan's moiety. It lies on the surface of type 2, acting as an interfibrillary glue. Type 10 is associated with calcification of cartilage. Disruption of the collagen network is a key factor in developing of osteoarthritis. Collagen forms the cartilaginous framework and is responsible for the tensile strength of cartilage. Regarding aggrecan. Aggrecan is highly glycosylated protein induced primarily in chondrocytes. A single aggrecan molecule consists of protein core and numerous highly charged glycosaminoglycan GAG side chains. In normal articular cartilage, many aggrecan molecules bind to a chain of hyaluronan, and this interaction is stabilized by separate link protein. Aggrecan molecules fill most of the interfibrillar space of the cartilage matrix. They contribute about 90% of the whole cartilage matrix proteoglycan mass, whereas large non-aggregating proteoglycan contribute 10% or less, and small non-aggregating proteoglycan contribute about 3%. This figure shows the type of proteoglycan. Proteoglycans are one of the glycoproteins composed of a core protein with one or more covalently attached glycosaminoglycan GAG chain. There are various proteoglycans classified in terms of which and or knew how many GAG are attached. The decorin is a single dermatan sulfate or chondroitin sulfate chain by glycan, two dermatan sulfates and or chondroitin sulfate chain. Versican is a dozen chondroitin sulfate chain, and aggrecan is many chondroitin sulfate chain and some keratin sulfate chain. Proteoglycan, 10 to 15% of wet weight, produced by the chondrocytes and secreted into the matrix. Half-life of 3 months. Responsible for the compressive strength of cartilage. It serves to trap and hold water to regulate the matrix hydration. The proteoglycans are negatively charged due to the presence of carboxyl and sulfate groups on the glycosaminoglycans, and so confer a net negative charge on the cartilage extracellular matrix. As a result, cartilage is highly hydrophilic with a tendency to trap or absorb fluid or swell to maintain the mechanochemical equilibrium. You will see a variation of this figure. This is among the common exam question you'll be asked to draw these proteoglycans. So aggrecan from a large chondroitin or the sulfate proteoglycan family combines with hyaluronic acid to form hydrogel, essential for resistance to compression forces in the articular cartilage. The proteoglycans of articular cartilage are complex macromolecules consisting of a linear protein core to which are linked long-chain polysaccharide moieties, glycosaminoglycans. The glycosaminoglycans are polyionic in charge based on the regular occurrence of carboxyl groups and sulfates along the macromolecules. Again, this picture illustrates the structure of an Aggrecan-type complex proteoglycan. Please get familiarized with this picture. The standard model for the steric structure and chemical configuration of proteoglycans, as proposed by Matthew and Lozazide, is that of a test tube brush, in which the central core of the molecule consists of a linear protein, and linked to it and radiating from it at right angles are numerous glycosaminoglycan chains, stiffly extended in space. Again, this figure shows the aggrecan and proteoglycan aggregate structure. For A, aggrecan, a hybrid proteoglycan consisting of a core protein with three lobular regions, the keratin sulfate and chondroitin sulfate. B. Proteoglycan aggregates composed of a central hyaluronan filament with numerous aggrecan molecules bound together. Yay! We have reached to the end of this video. See you in the part 3 video. Take care and bye.